Let's continue looking now at the DNA molecule. The first thing we need to know is that DNA is in fact a polymer that has monomer units referred to as nucleotides. So think about some other polymers we're familiar with. For example, cellulose. Cellulose is the polymer, glucose is its monomer. So with DNA, DNA is the polymer and a nucleotide is its monomer. Now, nucleotides in DNA are slightly more complex than what you've been used to because they themselves are made of 3 subunits. So where glucose is just a single unit that joins together to make your cellulose polymer, nucleotides have 3 important parts of their structure when we talk about them as monomers. Now, it's not too complex because if you thought about glucose, it would be fair to say that glucose is made up of, carbon atoms, hydrogen atoms, and oxygen atoms. And so we sometimes refer to carbohydrates as CHOs because we know they're made up of 3 smaller parts. Well, that's true of a nucleotide. But the nucleotides themselves are made up of a sugar, deoxyribose, a phosphate, phosphoric acid, and a base. Now the bases are adenine, guanine, cytosine, or thymine. We tend to abbreviate these by using their first letter, A, G, C, or T. Now you do need to be familiar with these names. You need to know that those 4 bases are in fact the bases that you will find in a nucleotide and ultimately in DNA. Now it's the nucleotides that join up to form the polymer of DNA. Let's have a look at a schematic representation over the page, acknowledging that each DNA molecule will contain from 10 to 100 million nucleotides. So here's where we see that the DNA molecule is a vastly more complex molecule than something like cellulose, where we're talking about thousands of monomers. Here we're talking hundreds of millions of nucleotide monomers. So let's have a look at a nucleotide. Here's an example of a schematic representation of a nucleotide. You have a phosphate group, you have that bonded to a deoxyribose sugar group, and attached to the deoxyribose sugar group is a base. Now if we carry on looking at this at a specific example, on the right hand side of screen you have P, indicative of the phosphate group. It's bonded now to your deoxyribose sugar. Can you see how that's a pentose sugar? And then up the top right we have an example of a base, and we'll have a look at which base that is in a moment. But there's a schematic representation on the left and an actual example on the right. You'll be pleased to know that the Board of Studies requires that you can represent a nucleotide in that schematic representation on the left. You don't need to ever be able to draw the example on the right, though you would need to be able to recognize the example on the right as an example of a nucleotide because it's composed of a phosphate, a sugar, and a base. Let's keep going and have a look at the polymerization process. Figure A there, moving down the page, is a schematic representation of a segment of DNA because you can see that you have the polymerization of the nucleotide monomer. So you have a phosphate sugar backbone is what we call that. See how it looks like it's stepping down? You've got your orange phosphate attached to your sugar, attached to your phosphate, attached to your sugar, attached to your phosphate, attached to your sugar, and on it goes up to 100 million times. Attached to each sugar is a base, and the base is hanging in the middle. We're going to see that it's that base that hangs off the sugar phosphate backbone that will give rise to what we recognize as our double helix. At this stage, we're actually seeing a single helix, only half of the DNA molecule. Diagram B is showing you a more accurate representation. Have a look at the phosphate sugar backbone. Phosphate group joined to the pentose, joined to the phosphate, joined to the pentose, and on it goes. And attached to each of those sugar groups is a different base. We're going to look at the bases more closely in a moment. So figure A is your schematic representation that draws out your sugar phosphate backbone. Figure B is an example of an actual small segment of DNA. Figure B, for example, is a segment that is only 4 nucleotides long, or 4 nucleotide monomer units long. So it's really just a segment. This would carry on up to about 100 million times for a real DNA molecule. Let's have a closer look now at the bases. Here they are: adenine, cytosine, guanine, and thymine. They all have some fairly similar characteristics. The most important thing for you to recognize in forensic chemistry is what we call base pairing. When we talk about base pairing, we're talking about the formation of hydrogen bonds between base pairs. If you look at the bottom left of screen, you can see that guanine and cytosine, due to the way that they can line up, can form 3 hydrogen bonds that pull these 2 sugars together in a pair. These 2 bases together in a pair. On the right-hand lower side of the screen, adenine and thymine can only form 2 hydrogen bonds with neighbouring nitrogen molecules, nitrogen atoms. Now remember, hydrogen bonding can only exist when you have hydrogens in a neighbouring molecule coming into close contact with either a fluorine, nitrogen, or oxygen atom. On the screen you can see that we have nitrogens and oxygens present in neighbouring bases. Therefore, if hydrogens attached to another base come within the local vicinity, they will be strongly attracted, forming a hydrogen bond about 1/10 the strength of a normal covalent bond, which means that these bases want to pair up. They get very sticky. And because of the actual three-dimensional shaping of these bases, guanine and cytosine will always pair together because they can form 3. Adenine and thymine will always bond together because they can form 2. You will never get pairing outside of these 2. So for example, adenine will not bond with cytosine. Adenine will only go with thymine. Cytosine will only go with guanine. How do we remember this? Well, I actually refer to it with a little rhyme. It's not even a rhyme, it's fairly lame, but I think in my head, at GC. Where do I need to go? I'm going to be at GC. Why do I think that? I have no idea, but I spell a little word, at. Oh, so A goes with T. And what's left over? G and C. If you want to know the origins of GC in my thinking, you can come and see me. I'm not going to put it on a video. So we carry on. There are our 4 bases. Remember, at GC, A always goes with T, G also always goes with C. For the forensic chemist studying the Year 12 course, that's an important point. Those are the 2 pairings you will find. You could well get a 1-mark question that simply says, I have adenine in the DNA molecule, what will it pair with? And you'd have to go, okay, A always goes with T, it will pair with thymine. You do need to recognize these structures as the bases, but you don't need to remember how to draw them or even their molecular formula. Well, DNA is famous for being a double helix. What we're going to see now is that it's the base pairing that forms the double helix nature of the DNA molecule. Here is a representation of the double helix. You have 2 strands that are single strands of DNA, but here you can see that hydrogen bonding exists between base pairs that holds 2 individual strands of DNA very tightly together. You can see the hydrogen bonds being indicated between the pairs, and A is always bonded to T, G is always bonded to C. We've got at GC. So the electrons of the H atoms are attracted to the very electronegative atoms of O and N in the neighbouring bases, and so this holds the paired DNA strands together in this double helix arrangement. Schematically, you can just represent it like this. You've got your sugar phosphate backbones, and you have a base attached to each sugar. There's 1 helix on the left and 1 helix on the right. Whatever base happens to be hanging off the sugar, it will pair up with its favourite base pair on the opposing sugar on the other strand. So at the top you've got T bonding with A, you've got 2 hydrogen bonds there. Notice how G and C have 3 hydrogen bonds, and then the lowest one again you've got A and T. You can invert the A and T as can be seen here, but again remember you only ever pair A with T and G with C. So in reality this helix, the helixes twirl around each other due to the nature of that triple hydrogen bond versus the double hydrogen bond, and it's a little bit like those old school telephone cords. You know, if you pull those out and then let them spring back together, they tend to coil around one another, and that's a bit like how DNA exists. Now DNA in your body is the building block of life, and along the strands of DNA are these specific coding regions. In other words, there's a series of base pairs that are recognized as areas that code or sequence for different things. For example, you might get this sequence of bases ATT, CT, GG, CG, CT, ATT. Now I've just made that up, but that's a possible sequence of bases on one of the helixes. And this could be a code for a gene. Now genes are responsible for coding for amino acids, which we saw in the introductory video, which means that this section of your DNA helix could well code for a gene, and that the gene causes amino acids to be produced in the cell, which then will give rise to proteins. Now each DNA molecule contains thousands of genes. Each of these genes will code for specific characteristics of the organism. Now, when we look at human beings, the reason we're all so very similar is that 99% of our coding DNA is identical. Now this makes perfect sense, doesn't it? Different genes are going to express different things in the organism. So the gene for eye colour and hair colour and skin texture and finger length and all these other kinds of crazy aspects of your body, that is very similar to all the other human beings. For example, you've got a liver and I've got a liver. That's because we have areas in our DNA that code for the build-up of the cells in our livers. And because we're so similar, or the reason we're so similar is because 99% or 99.9% of that DNA coding region is identical. So that's not actually very helpful for us when we start to think as a forensic chemist. If 99.9% of our coding DNA is identical, it's not much use in distinguishing between suspects at a crime scene. Because all you would do is you draw out all these gene coding regions and say, okay, the suspect is definitely a human being. And then you've got 6.5 billion suspects to choose from. But the non-coding sequences of junk DNA that's present in all DNA molecules is extremely different between organisms. What we do is we can cut the DNA up and we look at the length of the non-coding regions in a DNA helix that sit between coding regions. It's these non-coding junk segments that are of different lengths that are unique to every individual that forensic chemists use to differentiate between suspects. It's these bands that you see on electrophoresis gels that are actually bands of non-coding junk DNA that distinguish between suspects. We will look more closely now at this junk DNA in the next video.